Hello biologists, welcome back. We're going to continue our discussion on evolution. I want to remind you of why it is that you are now qualified to understand this subject better than other people. It's because a few lectures back we covered meiosis, and meiosis is the process by which we arrive at huge amounts of genetic variation in offspring. We also covered Mendel, and so you now understand the patterns of inheritance. We also covered DNA structure, so you understand how DNA works. You also understand how minor changes in DNA can produce really huge changes in the outcome of what that DNA makes. We also covered in the last lecture Darwin and how he arrived at his concept of natural selection, and this is really going to be our launching off point into population genetics. So what is population evolution? I want to bring up a term here, microevolution. We're not really going to use that term. However, a lot of textbooks do, and if you looked this up on the internet, you might find it. Microevolution really refers to something called allele frequency changes. And you all remember what alleles are. Alleles are all of the different variations of a gene for how to make a particular trait. The classic case is eye color. Obviously, there are many different ways of making eye color, and yet you, because you only possess 2 copies of your DNA, you only have 2 ways of making eye color, even though there may be many different kinds. So if you look in a population of many organisms, within those organisms on each of their DNA strands is going to be information on how to make all those different varieties of eye color. So microevolution is an old term that kind of describes this. The reason why I don't use this term anymore is because this term has been used in contrast to macroevolution. And really, there's no difference between microevolution, where you have genetic changes in a population occurring over small timescales, and macroevolution, which is the exact same thing occurring over very long timescales, geologic timescales. We'll get into that a little bit later. We also need to define what a population is. Population is really just all members of a species that occur in a particular area that have the capability of reproducing with each other. And I want you to remember that there is variation in populations. I showed you that in the last lecture. That is extremely important. However, it's not the only factor that determines a successful outcome for populations that are reproducing and making future populations. So in comes the subject of population genetics. It really is the study of the frequency of any gene in a population. For example, if you were to be sitting in a classroom right now, let's say we had 100 students, we would want to know what is the frequency of brown eye traits in the class. So how many of those 100 students have brown eyes? Let's say that in our case it was 80 out of 100. Well, then the frequency of the brown eye gene in our classroom would be 0.8 or 80%. What is the frequency of blue-eyed genes in that same classroom? Well, let's say for our own purposes here that it's 5% or 0.05. So that would be the frequency of the blue-eyed gene. Okay, so you kind of get the idea. When we're talking about gene frequencies in a population, we're talking about the percentage of the population that exhibits a particular kind of trait. And 2 scientists, who I'm going to introduce a little bit later, known as Dr. Hardy and Dr. Weinberg, independently came up with a way of measuring the change in these frequencies from one population to the next. And they basically asked the question, is a population evolving? That is, if I notice that 80% of a population has brown eyes, but then this population makes babies that exist in the future, and I look at all their babies, do their babies also have 80% brown eyes? If they don't, then possibly a change has happened and the population has evolved from one generation to the next. The nice thing about this Hardy-Weinberg equation, or Hardy-Weinberg equilibrium that I'm going to discuss later, is that it sets up what's called a null hypothesis. I want to bring you back to the very first lecture that we had where we talked about the scientific method to remind you what a null hypothesis is. A null hypothesis is really an idea that says there is no effect. There is no change. So if the Hardy-Weinberg equation is correct, then 80% of the population started out with brown eyes, and after they all had babies and we checked on that population in the future, that still 80% of the children, of the offspring, had brown eyes. In that case, the null hypothesis was true. No change occurred. Okay, let's move on. You have to start with some knowledge about the gene pool. That is to say, you have to go out into that population and first measure among that population how many of the students had brown eyes. In this case, we set it up as 80%. You can't know whether a population is evolving unless you have that prior knowledge. So we need to understand that. So for this next exercise, we're going to actually pick a trait within a species that is commonly studied for genetic tests, that being the fruit fly Drosophila melanogaster. This is your typical fruit fly that lands on your fruit and lays its eggs in it, and then you have to throw the fruit out because it tends to rot. However, in this fruit fly, there are a bunch of different ways that the fly varies, kind of like eye color in us. You know, there's many different eye colors. Well, in fruit flies, there's many different outward phenotypic appearances, what we call morphotypes. So there's many different morphotypes of a fruit fly. I'm showing you just some examples here. In this case, here's one that has your typical wild type red eye, and then they also have morphotypes that have kind of an orangey eye. There's also morphotypes that have a regular kind of body color, and then there's the golden colored ones. Here I have a black colored morphotype, and then we have this stubby wing variety here. So you can see there's many different ways that these flies can vary. Let's go ahead and just pick one type, and we'll call one the normal type. Okay, and if I back up, let's say this here is the normal type up here, but we're concerned with this one down here as well, the short wing type. And so let's assume that we start out with 100 flies, and we're just going to make the assumption that, let's say, 36 of these 100 flies are homozygous dominant. That is to say, they have the dominant genotype, the dominant allele, and it's represented on both of their chromosomes. So it is homozygous dominant. If 36 out of 100 of them are homozygous dominant, well, that's 36%. We can also say that let's assume there are 16 that are homozygous recessive, that are like this, the short wing variety. So out of the 100, 16 are homozygous recessive, which means 16% of them are homozygous recessive. So we can add this up, 36 plus 16, and the leftovers, the 48, let's assume that they are heterozygous dominant. So when you were to look at their phenotype, you were to look at their outward appearance, 48 of them would look just like these 36. It's just that we distinguish these 36 as being homozygous and these 48 as being heterozygous. Okay, so let's take these flies, these 100 flies, 36, 48, and 16, and we're going to add them all up. Certainly they all add up to 100%. Right, or 100, let's say. But I want to now look at the individual alleles within the entire population. We're looking at normal alleles and we're looking at short-winged alleles. We're going to use the letter L to distinguish this. So if you are exhibiting the normal body form, then you have at least a big L present. You can be either big L, or big L, little l. That is, you can be the homozygous dominant or you can be the heterozygous dominant. Over here, the short wing variety has two small l alleles, and so this would be the homozygous recessive case, where these 16 individuals would look like this, and these 36 individuals would look like this, and the 48 individuals would look like this. So in this population of 100 individuals, we want to ask ourselves how many big L alleles or little l alleles are present in the entire population. Not in individual flies, but in the entire population. So here's what we have to know. Let's go ahead and start with the big L allele first. If you are the homozygous dominant case here, big L, how many of these 36 individuals, or I should say, how many of these big L alleles are present in the 36 individuals? In this case, we can do it like this. Remember, there are two L alleles, two big L alleles in this homozygous dominant individual. And since there are 36 individuals, we can actually calculate it out as 72 actual alleles in the population that are big L, that are coming from this big L individual. Now we have 48 individuals that are heterozygous, right? They are like this, big L, little l. So if this individual, if all these individuals, these 48 individuals, are contributing only one big L to their offspring, then it would be 1 big L times 48, which would be 48. Now, the homozygous recessive case here, there are no big L's present in these individuals, so they're not contributing any of them. So if we add these up, we get 120 big L alleles that are present in these 36 plus 48 individuals, right? So let's do the same exercise now for the homozygous recessive case. We're concerned now with the little l alleles. Well, for the little l alleles, the homozygous dominant individuals are not contributing any because they don't have any in their DNA. However, the heterozygous dominant individuals at least have one masked little l allele. So 1 little l times the 48 individuals means that 48 of these little l alleles are contributed to the next generation by these individuals. Down here we have the homozygous recessive individuals, which have 2 little l alleles on each of their chromosomes. And since they have 16 individuals that are like this, then it's 2 little l alleles times 16 individuals, which is 32 total little l alleles. If we add these up, we get 80 little l alleles. Now, if we add up the 120 here and the 80, notice we get 200, right? 120 plus 80 is 200. Now, why is that? Well, remember, we have 100 total flies, but each fly has 2 sets of chromosomes. And that means that we should have a total of 200 total alleles in these 100 individual flies. Of the 200 alleles, 120 of them are big L's and 80 of them are little L's. This is really kind of critical because it means that the dominant allele, that big L, is more abundant in the overall population than the recessive allele, this little l right here, which only has 80 out of 200, which is 40%. So let me back up here because you will be required to do this on the next midterm. I want you to be able to know that if you have 100 individual flies, that really those 100 individual flies are represented by 200 total alleles because each one of these 100 flies actually has 2 alleles, right? In some cases, those 2 alleles are a big L, big L. In other cases, the 2 alleles are a big L, little l. And in the last case, those 2 alleles are a little l, little l. So if you were to calculate the number of big L alleles in these 200 allele positions in the 100 flies, it would be 120 using these numbers here of 36 homozygous dominants, 48 heterozygotes, and 16 homozygous recessives. And you need to be able to do this for the dominant allele and also the recessive allele. And if you take these numbers, 120 over the total allele count, which is 200, and the 80 over 200, this is where you get 60% of the big L alleles represented in the population and 40% of the little l allele represented in the population. So if you can do that, then you'll get this problem right on the exam. So these here, this 0.6 and 0.4, are the frequencies of the alleles in the sperm and eggs of this population that will give rise to the next population. You can use this information in a Punnett square in a way that is very similar to what we did in Mendel. However, there is a difference here. In Mendel, we're talking about a male and a female, an individual male and female mating, and we did the Punnett square to figure out what the frequency of outcomes would be for each of their offspring. In this case, we are talking about not a male and a female. We're talking about all males and all females in the population. So in this case, we're going to set up our Punnett square. Notice I've kind of put it sideways. It doesn't really matter. You can do it any way you want. But to calculate the allele frequency outcomes, we need to take those frequencies that we mentioned in the last slide, 0.6 for the dominant and 0.4 for the recessive, and we're going to set it up in order to figure out what percentage of our population will represent each of these allele combinations. Okay, so we set up the heterozygote case, big L, little l, and big L, little l here. Well, these two big L's are going to make the homozygous dominant case. This one here, big L, little l over here, and big L, little l means that these two are going to be the big L, little l allele combination. And this one here is going to be the little l combination. Now, when we were doing Mendel, we assumed that there was an equal chance here of having a big L and a little l. We called it 50-50 and 50-50 over here, which means that there was a 25% chance of having each one of these outcomes. But when we do it for a population where we're considering all individuals, we can't necessarily assume 50-50. In fact, we have to set it up where it's 0.6 for the big L's and 0.4 for the little l's, because these ones are more common in the population. So if we continue with this cross, then these are the numbers we get. 0.6 times 0.6 is 0.36. 0.6 times 0.4 is 0.24, which is the same over here. And 0.4 times 0.4 is 0.16. In other words, because there are more big L alleles than there are little l alleles, it is not going to be a 25% chance for each one of these. It's going to be skewed. In fact, there's a 36% chance of getting this, a 24% chance of getting one of these, and a 24% chance of getting this, and a 16% chance of getting this. Okay? So we do this because these are not equal in the population. And we figured this out by measuring how many of them, like in our initial example, how many of the class have brown eyes. If 80% of the class have brown eyes, it's not going to be an equal chance of having brown eyes versus blue eyes in the class, right? So we have to skew the numbers. If we take this L times L, which is L squared, and big L times little l, notice we have two of them, so it's 2 big L little l's, and then little l squared, the equation looks like this. So this adds up to the future population. If we add these numbers up, they all add up to 100%. 36 plus 24 plus 24 plus 16 adds up to 100%, or we just say 1 here. So L squared plus 2 big L little l plus little l squared is the exact same thing as the Hardy-Weinberg equation in your book. P squared plus 2pq plus q squared equals 1 is exactly how we get this equation over here that is arrived at by doing a simple test cross. Okay? Make sure that you review Hardy-Weinberg and you understand how it is that we arrive at this equation here, because that's really, really important. Hardy-Weinberg equation really is a null hypothesis. It tells you what happens in a population if there is no evolution taking place. Let's take a look at that now. Let's assume, for example, in our blue-eyed versus brown-eyed example, let's assume here that 80% of a population has brown eyes and only 20% have blue eyes. Well, that is different from the assumption we made when we did Mendelian genetics. In Mendelian genetics, you had one male and one female, and we were interested in the chance of having one allele type versus another in the sperm or eggs. In that case, we assumed that it was 50-50. Right? It was 50-50 when we did our Punnett square crosses. However, in this case, we're not considering one male and one female. We're considering all individuals in a population, in which case it's skewed. In this example, 80% of the individuals have brown eyes and 20% have blue eyes, and therefore the outcome of what you see in the offspring, in the gametes, is going to be 80% chance of being brown eyes and 20% chance of being blue eyes. So if you started out with that ratio, then that population had a whole bunch of babies, and all of those babies exhibited the same ratio, then no evolution has taken place. So let's take a closer look at the example that we used. Here's Dr. Hardy and Dr. Weinberg. Again, they didn't know each other necessarily. They discovered this kind of independent of each other. But in our example, we started out with 36% or 36 individuals that had the homozygous dominant condition, big L, big L. 48% had big L, little l, they were the heterozygous condition, and then 16% had the homozygous recessive condition, little l, little l. Now, when we ran this through our Hardy-Weinberg equation, we plugged these numbers in, 0.36, 0.48, and 0.16 into this equation right up here. After one generation, we ended up with the exact same percentages and the exact same number of individuals with those features. So if the beginning population had these percentages and the ending population had these percentages, then we say that no evolution had occurred. They have the same frequency before and after. The question is, what if you go back into a population, let's say 20 years later, after enough generations have transpired, and you find that the percentage here is not the same as what it started with? Well, then you can conclude that the Hardy-Weinberg equation has been violated, and therefore evolution has in fact occurred. So the Hardy-Weinberg principle becomes this null hypothesis that basically states that the population will not evolve. That is, the allele frequencies will not change as long as the following 5 conditions are met. So these are really important now. Pay close attention to this. First one, there can be no mutations. That is, you cannot have any DNA errors in order for Hardy-Weinberg to be upheld. That is to say that no evolution has taken place. This is really critical, and we're going to talk about the importance of DNA mutations for evolution in the long run. But if no evolution is taking place, the only way for that to happen is, number 1, to have no mutations, number 2, to have no gene flow, and I'm going to talk about what gene flow is in a couple of slides here. That is, individuals cannot be coming into your population or leaving your population, because that would end up changing the gene frequencies. The term that we use for that is immigration and emigration. Number 3, you have to have random mating. That is, individuals have to be wandering around mating with each other with their eyes closed. Now, we know that doesn't actually happen. However, in order for no evolution to take place, the mating has to be completely random. So we'll come back to that one as well. Number 4, no genetic drift. You have to start with a large population, and we're going to explain the difference between a large population and a small population and how it relates here to genetic drift in just a few slides. And lastly, no natural selection can be occurring. That is, the environment cannot be favoring one genotype over another. What is the likelihood that all 5 of these conditions are going to be met? That there's no mutations, no gene flow, all mating is random, no genetic drift, and no natural selection occurring? Yeah, the chances are pretty darn slim. In other words, just by any one of these things actually happening can change the gene frequencies in a population from one generation to the next, means that populations tend to evolve all the time. Okay, so continuing on with this, meeting all 5 of those conditions is totally rare. At least one of those factors, if not all of them, eventually are going to influence the frequency of the alleles that you see in a population from one generation to the next. This is exactly the definition of microevolution. Microevolution, there's nothing magical about it. We're simply talking about the change in frequencies of genes over time. That is to say, today there's 80% of the class that has brown eyes, but next week only 75% of the class has brown eyes. If that change happens, then we say that evolution has occurred. And this is exactly why Hardy-Weinberg, kind of in a backwards way, describes all of the factors that can actually cause evolution. Okay, so let's revisit those again. I want to remind you, these are the 5 conditions that actually speak about evolution, that actually allow evolution to occur. Number 1, mutations. Anytime you have mutations in a population, that essentially is the the necessary ingredients to have genes change over time. Number 2, gene flow. Gene flow, bringing individuals into a population or letting them leave. Non-random mating is number 3. That is, individuals choose who they want to mate with, and that ends up changing the frequency of genes over time. Genetic drift. That is, populations tend to change for reasons that have to do with, actually a few slides will mention, genetic drift. Number 5, natural selection. All those great things that Darwin talked about are actually occurring. That is, particular genetic traits are selected for and others are being selected against. Okay, so let's revisit each of these 5 conditions so that we can view them in all of their gory details. Number 1, genetic mutations. Again, I've kind of referred to this as the raw materials for evolution. But what ends up happening is when you have a slight modification in the nucleic acid sequence of DNA, whether you're accidentally adding DNA, subtracting DNA, or substituting the wrong nucleic acid in DNA, mutations end up introducing variation into a population that can eventually be inherited by offspring. Now, the variation can be adaptive. That is, it can help individuals adjust to new environments. However, it's important to understand that not all mutations are favorable. In fact, the vast majority of them are not. Like in this case right here. Obviously, this poor lamb with 5 legs is not necessarily as fit as a lamb with 4 legs. But on occasion, you can have a genetic mutation that will at least not kill the organism, and on occasion can even be a benefit. Now, how do you get genetic mutations? There's lots of ways to doing it. Remember when we were talking about DNA replication? DNA replication was really all just about photocopying DNA, but it's very easy to have an error, a duplication error, where a whole segment of DNA that as it gets copied accidentally gets copied a second time. So in this way, you have dramatically changed the DNA. You've changed its reading frame. Remember about anticodons and how they put proteins together? Well, this totally screws up the protein that might have been made by this section here. Number 2, you can have inversion mutations. You can have whole sections of chromosomes that actually flip. Think about the reading frame again. In this case, this whole section of DNA, think of it like a chapter, gets read backwards. What kind of proteins are made when you read a reading frame backwards? Well, some really strange ones, I'm sure. Now, insertions and deletions. By simply inserting or deleting a nucleic acid in one place, it changes or shifts the entire reading frame. So in this case, where the reading frame would normally read histamine, 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 etc., all the way down, all these amino acids in a row, just by accidentally removing this A right here in the DNA sequence, changes every single amino acid after that point in the reading frame. Same thing here with insertions. If you have this same kind of starting position, adding an A into the DNA sequence totally changes everything after that. So with a very simple change in DNA, you can get some very dramatic changes in the protein sequences that come from that. Okay, number 2, let's talk about gene flow. This is movement of new genetic information either into or out of a population. Let's do into first. Alright, what are you hearing there? Okay, those are flies. Let's imagine a fly population here. These are a whole bunch of flies, and all of a sudden, in comes a weird fly. This is a fly from next door or from over the river. And it comes in and begins to mate, and hey, maybe having this morphology is actually beneficial to the population. If that's the case, then over time, it's going to increase in frequency. Kind of like that. However, movement into a population is important, but movement out is also important. Imagine that this is your population here, and just by random chance, maybe a rainstorm comes through or a boulder drops on this population, and in no time, a whole segment has moved out. And now the gene frequencies, that is the frequency of the black gene versus the golden gene, completely changes within a matter of a single generation. So this is how gene flow can change the frequency of alleles in a population. Number 3 is non-random mating. Okay, you all know it to be true. You guys don't just mate with your eyes closed, right? You at least want to look at who you're mating with. So this falls into 2 actual categories. The first one is assortative mating. This is when people of a particular phenotype selectively mate with other individuals that also have that phenotype. Think of it like tall people tending to find other tall people attractive, right? When they mate with each other, they're going to produce tall offspring in general. That's assortative mating. The other one is sexual selection. In this case, individuals enhance the attractiveness of themselves by their genes, by their DNA, in a way that increases the likelihood of their reproductive success. And really what it comes down to is access. It doesn't matter how brilliant you are, how smart you are, you could find the cure for cancer. If you're not all that attractive, people aren't going to talk to you in the bar. So that's what I mean by sexual selection. Let's take a look at a couple of examples of that. Assortative mating, right? Short people tending to find short people attractive, tall people tending to find tall people attractive. So these are examples of assortative mating. Now, sexual selection, these are 2 great examples of how organisms have, over evolutionary time, increased their sexual attractiveness. The feathers in a peacock. Obviously, that is energetically expensive to grow those feathers, and yet it persists because it increases this male's chance of reproducing. Same thing over here with this fiddler crab, which has a gigantic claw, which is so huge it's too big to even pinch things with. And yet, just by waving it around, they end up attracting mates. So why would these traits evolve? Simply because it increases the chances of mating by these individuals. Now, why are these traits attractive? Well, clearly, growing these big horns like this serve more than just a function of warding off predators. You don't need gigantic horns like that to do that. So why grow them so large? And in this case, colors like blue and red, those are expensive colors to make. Why spend all that energy in doing that? Well, it's very simple. If you are trying to show a mate, a potential mate, something unique about yourself, then the message that you're going to convey is, number 1, look at me. I am so strong, I can actually put out all this extra energy for these really weird features I have on me, and I can still survive. I am a strong survivor. And also, I have the ability to acquire extra resources. I'm so good at getting food, you definitely want to mate with me, because your offspring are going to acquire my characteristics of being able to find food. And I can do that even though these things that I'm making are expensive. So it's kind of interesting. That has evolved in lots of animal systems, including humans, you guys. You are not immune to this. How about this? Yeah, that's right. I know half of you out there are going to be like, wow, that's pretty attractive. And, you know, the other half of you are probably saying, what's so attractive about that? I got more than that guy. Anyway, the question is, why is this guy attractive? Why is it that Brad Pitt is attractive? Why is it that Pancake Boy over here is not attractive? The point being that even in you, sexual selection exists. You don't know why. Somewhere in your brain, it's telling you that one of these guys is attractive and the other's not. So let's go ahead and take a pop quiz. Who's more attractive, Brad Pitt, Pancake Boy, or who knows? What's more attractive? Well, obviously, it's in the eye of the beholder, but for the most part, sexual attraction is very important in humans. And the question then is, why is Brad Pitt sexier? Is it because of his body, his face, his mind, or his bank account? Well, probably in this case, all of the above, right? But, you know, if it comes down to reproductive success, and really these traits evolve because of sexual selection, having greater access to females, maybe that might, in this case, Brad Pitt, more access to females, but it can be the other way around too, right? Female attractiveness attracting more males. Well, it might explain why this occurs, right? I mean, after all, Donald Trump isn't all that attractive of a guy, is he? However, he exhibits other survival traits that might be attractive to other women who, in other circumstances, had he not been a wealthy man, wouldn't give this guy the time of day. It also might explain a few other behaviors. Like, how about these two guys? Yeah, what kind of evolutionary message are these two guys conveying to the ladies? Well, yeah, what's the evolutionary significant message behind this? How about it's this? Pardon me, miss, but should you choose to reproduce with me, then your offspring will have the greatest chance of survival. I mean, the guy on the right there is conveying this message of, I'm so tough and I'm so superior to everybody that I'm actually a target. People want to kill me and I therefore have to wear a bulletproof vest wherever I go. And the guy on the left is like, ladies, you definitely want to be with me because I am so rich I can flaunt it and even put it in my hat. Wow, that's pretty attractive. But at least you understand how this works. And in fact, in certain animals, since they exhibit certain behaviors, but they also exhibit certain outward appearances. These are all 4 really good examples of males that go far and beyond creating these really showy outward appearances to increase their mating opportunities. Okay, let's go on to number 4, genetic drift. We were talking about population size and how it's so important. Well, it's more likely for small populations to have dramatic changes within them compared to large populations. And I want to use M&Ms as a really good example of this to kind of demonstrate it. Let's say that you have 10,000 M&Ms. I don't know, what is that, a 5-pound bag of M&Ms? Something like that? All right, let's assume that the percentage of each of the 6 different colors in the M&Ms is equal. Of course it's not, it's actually skewed a little bit, but for this example, let's go ahead and assume that they were the same. So if we're 6 out of 100%, that means each one has to be 16.7%. Let's just say it's like 16 to 17 individuals out of 100 for each of these. Now, if you were to close your eyes and reach into this 5-pound bag of 10,000 M&Ms and you randomly grabbed 100 individuals, your chances would be pretty good that of each one of these that you grabbed, you'd get about 16 or 17 of each color. Does that make sense? Yeah. Well, but what about if you took one of these fun size, the ones that they hand out at Halloween, the one that's where there's only like 60 M&Ms in them? What are your chances that if you were to reach in to this bag of 60 M&Ms and you grab just 6 of them, how likely is it that you would grab one of each color? Probably pretty unlikely. That's because as the population gets smaller, the chances for random effects to take place to dramatically change a population go way up. The smaller the population, the greater the chance that the gene frequencies are going to change just by random chance. So when a population is small, this can easily happen. That is, if you just reach into a small population and pull a few individuals out that are successful at reproducing, overnight it can very quickly change the gene pool. So you very quickly get a loss of potential alleles just by random chance. Here's a really good example of it. Let's say if you have really nice diversity here, but just by random chance all the individuals over here died, well, your gene pool dramatically takes a hit and becomes far less diverse in just one generation's period of time. And this is precisely why it is so important to preserve genetic diversity. And I'll explain why in just a second here in this next slide. But one particular variety of this genetic drift effect is the bottleneck effect. The bottleneck effect occurs when a population's size is dramatically reduced. For example, you go out there and you overfish a population. Instead of there being 1 million fish out there representing a whole bunch of different alleles at all their various positions on their DNA, you dramatically reduce a population down to only a few dozen fish. Well, now, just by the very fact that you have fewer fish, you have significantly dropped the genetic diversity. This is true of cheetahs. Cheetahs are a great example of this, where cheetahs used to be very plentiful, many tens of thousands of them on the plains of Africa. However, they've been so dramatically poached and killed off that their population dropped to a mere few hundred. And now we're trying so desperately hard to mate them and cause them to reproduce, to try and increase their numbers. But even as we increase their numbers, they still have the genetic diversity of only a few individuals. Let me give you a classic example of the bottleneck effect, the northern elephant seal. This is my little vertebrate here, my little vertebrate offspring, who is pointing out elephant seals up in San Simeon Beach in Central California. Now, you know, these seals actually have a pretty darn big population now. They're actually numbering somewhere around 40,000. So you might think, oh, they must be in good shape, right? Well, not necessarily. You see, way back when, about 80 years ago, 100 years ago, the elephant seal was almost hunted to extinction. In fact, only 20 individuals remained at that time. We thought actually that we had fished them all out, but it turns out we hadn't. There were at least 20 individuals left, and the population has since rebounded. Actually, there's 30,000. We're probably closer to 40,000 now. However, this rebound of 30,000 individuals came from the 20 individuals that survived the massive slaughter. And we know this because if you do some genetic tests of this population that's out here on the beach, you find that they have no allele variation at 24 different loci on their DNA. That's like saying, in the past, there used to be 40 different ways to make eye color. However, you have so dramatically fished them down that the only ones remaining are the ones with brown eyes. So that's the same thing. There used to be many different ways to make eye colors. Now there's only one way, brown eyes. So we see that in the northern elephant seals. So they have very, very low genetic diversity. And what we will find later when we get into the ecology section is that very low genetic diversity means that you are very vulnerable to extinction. So even though the northern elephant seals it's numbering in the tens of thousands now, they are still at risk of extinction because they have extremely low genetic variability. Okay, there's another effect here with genetic drift. It's called the founder effect. The founder effect is when members of a population depart and go and reseed a new population. Classic example of this is the Amish. The Amish, coming from Europe, a very small band of people came over from Europe and established a settlement in the New England area. And they happen to exhibit a particular genetic trait called polydactylism. That is, having more than 5 fingers. It's a rare genetic trait in the population at large, but because it was in some of the original Amish that came over, and they didn't tend to interbreed with the humans around them, but with each other, it provided an opportunity for this genetic disease to actually manifest itself in far greater percentage and far greater frequency than in the natural population. Here's another great example of the founder effect. Let's say, I want you to think about the genetic makeup of let's say the United States. It looks like this. Here's the ethnic makeup of the United States. It's about 66% Caucasian, 15% Hispanic, 13% African, 4% Asian, and 2% everybody else. All right. Now, think about the classes that you've taken at Santa Ana College or any other college. Was the ethnic makeup of your class, did it look like this? Probably not. In fact, it probably looks very different from this. So let's imagine this. What if a meteor came and destroyed most of the United States? Okay, most of the United States is now dead, except for a very small population right here at Santa Ana College. And you guys in Santa Ana College, you guys had to have the dubious task of trying to repopulate the entire United States. Now, does your genetic makeup, your genetic or ethnic diversity look like this? No, of course not. It looks very different. So it means that the population that you give rise to, very quickly overnight is going to look very different from the population in the past. So the founder effect kind of operates on this principle right here. The classic case of this, what if the class had to recolonize America? What would it look like? It would look very different. The classic case is Darwin's finches. Okay, I want you to think back to Darwin's finches. When you had an individual pair of finches, or a very small group of finches that managed to make it over to the Galapagos Islands, and then they radiated and actually changed the whole genetic nature of how they looked out on the islands relative to how the finches look on land, or on the mainland. Okay, now this last one is one that you're actually familiar with because we talked about it in the Darwin lecture, natural selection. Natural selection adapts populations to the current environment. Of course, we know that the environment is always changing, and so the frequency of genes is always responding to this environmental change. And there's a couple of different things that affect populations. The first ones being abiotic factors. Remember, anytime you see an A in front of something in science, it usually means not having it or not possessing it. So abiotic factors are those things that have nothing to do with biology. For example, it's too hot or it's too cold, too wet, too dry. All of these sorts of things are physical phenomena about the world around you that have nothing to do with biology. However, there are certain organisms that are going to survive, or certain traits that are going to allow you to survive this better than others. Then there's the biological factors. Competition, predation, parasitism. These are things that are facing populations too and cause them to change over time. We're going to get into both of these in greater detail in the ecology lecture. So natural selection, let's say that you have this phenotypic variability here that I'm showing you here in these mice. Let's say that, for some reason, there's an advantage to having this darker fur color over here just because the environment has changed in a way that favors that. Well, you're going to get a directional shift in the gene frequencies. And this sort of gene frequency shift is known as directional selection. In the same vein, you can also have a situation where the extreme phenotypes are selected for, like white or black, but not brown. The one here in the middle is actually selected against. If that's the case, then you have a gene frequency shift that looks like this, where the extremes are selected for. This is known as disruptive selection. Then there's the condition, the far more common condition, where the intermediate phenotypes are the ones that are the most desired, or the ones that survive most or best in the environment. And if that's the case, then the extremes tend to drop out in frequency, and the middle case, or the intermediate phenotype, is the one that increases in frequency. So these are three different modes of natural selection that we see often in nature. The classic case of the directional selection is the peppered moth. And we're going to cover this guy a little bit more later, but this is the peppered moth right here. You guys see the moth? Well, of course you do. You see this guy right over here, this black moth. But what you don't see, or at least what is probably hard to see, is this moth right over here. This moth is very well hidden because it blends in with the lichens on this tree trunk, right? Well, imagine being this bird. If you're a bird and you're going in here to try and eat moths, who are you going to eat first? You're going to eat the one that's very easy to see. You're going to eat the one that's black with the mottled background because he's the easiest one to see, right? Okay, so this is a selective pressure on this population of moths that increases the frequency of this mottled phenotype, and it selects against the black phenotype. However, we're going to talk more about the Industrial Revolution a little bit later, but we can at least mention it here. The Industrial Revolution in Europe belched huge amounts of soot and ash into the air once factories got going running on coal, especially coal. And it ended up killing all of the lichens on the outside of these trees in Europe. And when that happened, it also blackened the surface of the trees, and it created this really strange condition where the black moths were the ones that were more hidden from the birds. And so within a matter of just a few dozen generations, This morphotype right over here, the mottled morphotype, actually began dramatically decreasing in frequency, and the black morphotype began increasing in frequency. Now, the odd thing is that in Europe, once the Industrial Revolution started switching over to things that were a little bit cleaner burning, and also they began passing their environmental laws, you actually saw a return of the mottled phenotypes, which is the way it is today. The mottled phenotype is far more numerous today than it was just 70 or 80 years ago because of this. Another classic case of directional selection is horse evolution, where the original horses that lived in North America in the geologic past were very small animals. Hyracotherium actually was very nimble and small and was ideal for running through small forests with lots of trees, very tightly situated together with lots of roots. If you were a big horse like the ones we see today trying to run through there, you'd break your ankle. And so there was a very strong selection for very small body size. However, during the last few ice ages, the forests actually began receding here in North America, and it gave way to wide open plains of grass. And with wide open plains of grass, the situation facing horses dramatically changed. Now there were open area predators that would run down their food, and it became selectively more advantageous to be larger size, to have sturdier legs for running full on out at full speed. And so over time there was this evolutionary trend that we see very clearly in the fossil records towards a much larger, much sturdier horse. And this is our modern horse today, Equus. And directional selection actually has taken hold also in industry due to something known as antibiotic resistance. In this case, when you are trying to, in this case, kill bugs on a leaf, let's say that you have a field out there full of spinach, but you have a particular insect pest that's invading your spinach. Obviously, you want to kill them. Well, what a lot of people have done over the last 100 years is develop chemicals to spray all over your spinach to kill the bugs. However, you may not kill all of them. There may be a few that actually, even if they might get a little sick, they don't die. And these are the ones that get left behind and reproduce and make the next generation's bugs. And if you continue to spray, over time you're killing off those that are susceptible to your spray, and you're selecting for those that are not susceptible to your spray. And very quickly you end up with a whole bunch of bugs on your crops that are completely resistant to your spray. Same thing is true with antibiotics. If you take antibiotics and you don't do it properly, you can actually end up selecting for the bugs that don't seem to be killed by antibiotics. In the end, you end up getting sicker over time. We'll cover that more later as well. Let me talk about stabilizing selection now. Stabilizing selection is when the intermediate phenotype gets selected for, and then you end up narrowing the variation of phenotypes over time as the population adapts to a very stable environment. This is actually the most common type of selection, and the odd thing is that over time, as you reduce the variation of phenotypes, you actually end up putting your own population at greater risk of going extinct. And yet, it is the most common form of selection. Okay, now, disruptive selection. This is very interesting because the outcome of disruptive selection is going to be the launching pad for the whole next lecture. In this case, natural selection ends up favoring the extremes at the expense of the intermediate phenotypes. So it creates this division which ends up leading to very, very different phenotypes, and ultimately, this can lead to speciation, the creation of two unique species. And I'm showing you an example here of two snails, and one is left-handed and the other is right-handed. In this particular case, there is no intermediate phenotype to be selected for, and so it drives these two phenotypes apart, further and further apart, until they are so different that they can no longer successfully mate with each other. This is critical because this is how new species emerge over evolutionary time. Now, an important thing to keep in mind is, everybody kind of thinks of evolution and think, evolution is moving towards progress, moving towards something that's superior, something that's better. However, that is not the case. The question that I pose to you is, why is it that natural selection does not fashion perfect organisms? Remember, natural selection is a response to a changing environment. And so long as the environment continues to change, natural selection will never evolve towards any one epiphany or perfect organism. Also, selection can only act on the existing variation that you currently have. You can't just reinvent things just because you want to. Remember that slide I showed you before about the beavers? There's no such thing as a beaver evolving a chainsaw for a hand. Even though it might help that beaver, that evolutionary process can only act on the existing phenotypes within the beaver population. So evolution is also limited by historical constraints. You're not going to see beavers evolving wings just because it would benefit them. If the previous beaver population does not have the predisposition for wings, then you're never going to see wings evolve in that group. And, adaptations really are often a compromise. They are not the perfect solution. They're simply the best possible solution that happens to arise right now. Remember, it's not survival of the fittest. Remember, that was not a Darwin term. That was actually a term invented by an economist. So it's not survival of the fittest. It's survival of the not dead. If a trait doesn't kill you, then it will still get passed on. Also, chance, natural selection, and the environment all interact with each other to produce successful organisms. And what happens if natural selection occurs over many, 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 many generations? You can get huge changes. Really, really big changes. And we know these changes as speciation. Speciation really is the creation of new populations of organisms that are distinct breeders. That is, they breed with each other, but not with other closely related organisms. This is ultimately what has given rise to such an amazing array of organisms on the planet, such a huge diversity and abundance. And each one of these organisms is represented by a unique genetic signature, a unique combination of DNA that the world had never seen before. So this ends this lecture. I will see you all later.